Hi, my name is Winson Zhang. Today, I will be presenting the various applications of the current CRISPR technology, as well as discuss some of its limitations and ethical concerns surrounding this revolutionary gene editing tool. So, to start today's presentation, I briefly want to go over the previous gene editing tool before CRISPR technology, which is gene therapy. Gene therapy is the practice of introducing new genes into a person to modify their genes to treat or prevent a certain disease. It was one of the earliest gene editing technology that showed promise in treating diseases that have been long difficult to treat. However, it faced several challenges, such as limited accuracy, which could trigger genetic errors and unforeseen results, and a high up cost. One specific case is the case of Jesse Guy Singer, who tragically passed away from severe immune response to adenovirus vector. Enter CRISPR, or cluster regularly interspaced short palindromic repeats, a revolutionary technology that addresses the limitation of gene therapy with its unprecedented accuracy and speed. Before I talk about its current application, I wanted to briefly go over the basic mechanism behind why CRISPR is superior to traditional gene therapy. CRISPR was initially discovered by scientists as being a fundamental component to the bacterial archaea adaptive immune system. Scientists observed clusters of regularly spaced repeating sequences in bacterial genome. Between these repeats, they found a unique sequences of DNA known as spacers, which contain fragments of viral DNA from previous infections. The combination of these spacers and repeating sequences creates what is known as a CRISPR array, which serve as a memory system for the bacteria to prepare against future infections. During a viral infection, the CRISPR array is transcribed into a long RNA molecule, which is processed into individual CRISPR RNA, or cRNAs, that contains a unique sequences that correspond to a past viral DNA infection. This process is largely done by another RNA known as the trans-activating CRISPR RNA, or tracer RNA. Together, the cRNA and the tracer RNA binds to a Cas9 protein, a nuclease that acts as a scissor in the CRISPR-Cas9 system. This entire complex then scans along the viral DNA, with the cRNA serving as a guide for the Cas9 protein to locate the viral genome. For Cas9 to recognize and bind to a target, the presence of the protospatial adjacent motif, or PAM, is required to be adjacent to the target DNA sequences. If CRISPR detects a strand of sequences that matches the cRNA, and is adjacent to a PAM, the protein would then proceed to cleave the area, preventing the viral infection. And recently, in lab, scientists were able to combine the cRNA with the tracer RNA to create a single guided RNA, which then can be programmed to match specific DNA of viruses, which is what gives CRISPR its accuracy and speed in gene editing. Now that we covered over the basics of CRISPR and how it functions, I will now go into its various application. Start off with its application in human health. Due to CRISPR's ability to accurately detect a wide range of DNA sequences, CRISPR has been implemented in diagnostics. CRISPR-based diagnostics has successfully detected diseases such as tuberculosis, as well as malaria-causing parasites. It has also been used to detect non-infectious disease such as acute kidney transplant rejection. Other things that CRISPR diagnostics can detect include point mutation, protein, and small molecule. For example, it has been used to serve as a sensor for ATP. Moreover, CRISPR technology also provides potential solution in addressing the recent COVID-19 pandemic. For example, the SARS-CoV-2 detector assay delivers results in about 40 minutes and 95% accuracy. A technique called Pac-Man offers therapeutic solution by addressing the high mutation and recombination rates of the SARS-CoV-2. CRISPR has also been successfully implemented in clinical trials in gene therapy and cancer immunotherapy. The first CRISPR-approved clinical trial in the US was in 2018, where scientists modified T cells to target relapsed tumor that has no further treatment options. Since then, other clinical trials have been approved to use CRISPR, such as using CRISPR to delete PD-1 genes, which is a gene that disrupts T cells from killing cancer cells to treat the likes of esophageal cancer and invasive stage 4 bladder cancer. CRISPR has also been used in gene disruption, such as treating patients with sickle cell anemia by disrupting the BC111A gene to increase fetal hemoglobin levels. Beyond human health, CRISPR also finds itself having various applications in agriculture as well. CRISPR has proven to be useful in the field of agriculture because of its ability to easily manipulate the genome of plants. CRISPR has already successfully improved cereal crop yield by editing the genes of O. sativia pin5b, which controls panicle size, and O. sativia GS3, which controls grain size. It has also improved quality, such as increasing the amount of amylase in rice, as amylase is a beneficial prebiotic for digestive health. Besides improving the yield and nutritional value of crops, CRISPR has also developed disease-resistant crops, such as rice resistant to Xanthomus oryzae, a bacterial blight that threatens global rice production, and wheat resistant to biotrophic fungi, Plumeria gramis. CRISPR has also been used to accelerate the domestication of orphan and wild crops. Orphan crops often have favorable nutritional value and natural resistance compared to traditional crops, which could help address the rising food demand. Some examples include editing the genes of CLV3 and WS that control the fruit size of Solanum pinifolium, a tomato ancestry resistant to pathogenic bacteria and salt. Other orphan crops such as Dino intermedium, a perennial wheat relative that is more energy and water efficient than traditional wheat, as well as Solanum SPI, a potato variant that is tolerant to abiotic stress and contains high nutritional value, have all garnered agricultural interest and is looking to use CRISPR to remove undesirable traits such as low yield rates. However, despite all the benefits CRISPR has to offer in human health and agriculture, there are several limitations and ethical concerns surrounding this technology that needs to be discussed. Some current limitations to the current CRISPR technology includes the relatively high frequency of off-target effects. This happens when Cas9 protein cleaves at the wrong site, which could potentially lead to unforeseen consequences. One strategy to minimize the frequency of off-target effects is optimizing Cas9 variants, such as Cas9 nickase, to specifically reduce off-target effects. CRISPR has also experienced limitation in delivery methods. Ex vivo delivery requires an adequate amount of cells and retention of the modified cells once reintroduced, and in vivo requires extra caution and precision, which can increase complexity. While CRISPR diagnostic is powerful in detecting pathogen and other substances, the dependency on preamplification for detection below the femtomolar range increases complexity, cost, and reaction time. One of the most controversial usage of CRISPR is human germline editing. This is because it removes autonomy in decision-making for the individual being tested on, as well as the potential for unforeseen and permanent side effects that can be passed on to future generations. One example is the case of Chinese scientist Jian Kui-he, who modified genes in a human embryo to develop HIV-resistant babies. So now that we have talked about CRISPR limitations, I want to discuss where future research should focus on in improving the current CRISPR technology. Future research should focus on reducing off-target effects, improving delivery methods, and exploring new applications for CRISPR in environmental science and industrial biotechnology. Future research should also consider and evaluate other ethical concerns, such as the accessibility and affordability of this treatment, to prevent genetic inequality. The concept of designer babies will also need to be discussed, as it could set undesirable standards for future generations of parents to edit their descendants. Additionally, regulation needs to be established to ensure safe and responsible practice of CRISPR technology. By addressing these challenges and ethical concerns, CRISPR can offer solutions in various fields, from agriculture to human health and more. So, in conclusion, CRISPR-Cas9 is a groundbreaking advancement in gene editing, with its current application in diagnostics, gene therapy, agriculture, and more. While it offers many benefits and solutions to problems that has long been difficult to address, addressing its limitation and ethical concern is crucial for its safe and responsible practices. I hope I have given you a better understanding of the importance of CRISPR technology, and thank you for your attention today.